Our first speaker is Itai Yanai, who's going to talk about some transcriptional behaviors in mid-development in embryos in a number of different species. So it'll be quite interesting.
Okay, great, thank you. Hi, everybody. So in my lab, we study development and evolution, all through the lens of gene expression. And what I want to tell you about today is how we think we identified a universal aspect of animal development, and we call it the mid-developmental transition. And we show that when you study this mid-developmental transition, you really see how a particular organism defines its body plan. So let me get going. When you look at the tree of animal species, you see that there's different branches. And for particular branches, there's been called a name called a phylum. So a phylum of species is just a branch along this tree of animal life. And the kind of delineation of one branch from another has been according to body plan. So each phylum is supposed to have a particular body plan. And if you want to understand how that body plan is laid down, well, for that, you need to go to development. And already in the 19th century, German biologists have shown that you need to look at the embryo if you're going to understand the differences between different animals. And so it started with Carl von Baer. What Carl von Baer showed is that when you look at the way the embryos develop, the particular items of an animal, like the snout of a pig or the beak of a bird, all those particular things come later. And the general things are laid down first. Sounds obvious to us now, but someone had to say it. And Carl Haeckel, when he looked at the stages in mid-development, he recognized tremendous similarities. And those similarities he inferred to be tremendous evidence for evolution. They look similar because they had a common ancestor. These days, when we look at a set of species, we summarize it in terms of an hourglass model. So what's an hourglass model? You know, an hourglass has like a fat beginning and a fat ending, but in the middle, there's like a narrowness, right? So if you look at that as though it's development, where let's say that this axis is time, well, if you take species of a particular phylum, they could be very different at the beginning. Big eggs, small eggs, medium-sized eggs, like a chicken egg, a xenopus egg. And at the end, of course, they're very different, right? You look at a chicken and a fish and a pig, they're very different. But in the middle, you have this constriction. And why would you have that constriction of variation? Why would they be so similar? Well, our best hypothesis, and Harel Zaltz from my lab is going to talk about it soon, is that there is a lot of purifying selection that's acting at this stage. There's a lot of developmental constraints. And in particular, we know that the Hox genes are turned on at this period. Okay. So in my lab, we wanted to study this, not at the level of just the way the morphologies appear to us, but actually, to look under the hood and study the gene expression. So one thing that we did 4 years ago, and I actually reported it the last time I was at this meeting, was that we took 5 different nematodes, C. elegans and 4 friends, and we isolated embryos throughout 10 developmental stages. We monitored what every single gene is doing with a transcriptomic method. I'm being vague because it was microarrays, not RNA-seq at the time. And then for every gene, we had a plot like this. So for example, for T-box 43, we see that in C. elegans, the orange, it's low at the beginning, so there's not a lot of transcripts at the beginning of development. And then the gene gets turned on, there's lots of transcripts, and then it comes down. And that's conserved across the other 4 species. Isn't that cool? Well, using this data, because we have 20,000 of these plots, you can say, well, where's all the differences? Where's the conservation? And when you do that, you see that not each developmental stage is equally conserved across the 5 species. Some stages can vary. Some could be more conserved. And if you look at this the right way, like we did, you might see a kind of hourglass model. And that's nice because that means that not only at the level of morphologies, but also at the level of the molecular gene expression levels, you have an hourglass model that summarizes what kind of variation you see when you compare species of a particular phylum. And the same analysis was done also in a set of arthropods, and an hourglass model emerged, and also in a set of chordates and an hourglass model emerged. So what we wanted to do is now say, well, can we now compare different phyla? Can we say, not just within a group of species of the same phyla, can we now compare all phyla and see where the variation lies? How do specific phyla define their body plan? How does it happen? So this is a project that started 4 years ago. It just finished. It was just published. And it was really heroic work by a PhD student, Michal Levin, and a master's student, Leon Anavi, doing the experimental and the computational work. We had many other people working with us, but they really did the Herculean efforts of masterminding and leading it all. And what we did is we said, we're going to choose not 5 nematodes this time, so we're going to choose 10 species, but we're going to choose those species so that they have the greatest possible variation among them. All right, so how would you do that? If you obviously wouldn't choose a chicken and a pig because they're both vertebrates. That's a total waste, right? You've got so many other different kinds of animals to choose from. So basically, of the 35 phyla that are annotated on Wikipedia, many of those you just can't work with. You look at some of these phyla, it's just one species that no one's working on, so you can't get at that. Or it's like 2 species, but there's no genome. So it's really difficult. Anyway, we chose the 10 species, the 10 phyla that are the most species-rich, the most popular, and pick just one species from each. So we have a zebrafish, we have a ctenoform, we have a tardigrade, we have Drosophila, of course we have C. elegans, a sponge, a cnidarian, a sea urchin, a flatworm, a segmented worm, and it was great fun. What we wanted to do was, again, do a time course. So we isolated 70 embryos for each of these species. And the 70 embryos span the fertilization stage all the way down to the free-living or larvae. And for this, of course, we needed lots of help. So we teamed up with many labs in order to just get our hands on the animals, get our hands on the embryos. And it was really exciting to collaborate with all these people. And we learned a lot about the different customs and traditions of each field. Some fields are cutthroat, some fields are... So it was really interesting. And at the end of the day, once we had the embryos, there was a lot of work of how you process them now, because we don't want to just look at the embryos, we want to study them molecularly. And for 3 of the species, there wasn't a genome, even though I just said I'm so averse to not having a genome. In 3 cases, it was a really species-rich phylum, like the planaria. You got to tackle the planaria, but there was no genome for the particular organism we had to use that had a convenient development. So what we did is we made our own de novo RNA-seq transcriptome for 3 species. One of them was tardigrades, because this is before the genomes, I should say, were published. And we did it so it's not... okay, so when we did when we collected the data, each sample is a single embryo. And that's good because usually we take like 50 embryos and pull them together. So it's good, just one. But it's not as good as what we can do because we developed a method recently called cell-seq, which is awesome for doing single-cell RNA-seq. And by the way, one of the projects we're doing now is to take C. elegans, and you know how there's 558 cells at the end when it hatches? Well, if you add up all the cells throughout, it's 1,304 cells, and we're identifying the transcriptome of each cell. So we can do that using cell-seq, but for this project, it would have been too much. So we did it single embryo transcriptomics, and we developed tools for how to align the embryos, because in some species, you collect the embryos, like in sponge, but you don't know who's ahead of who. It's hard to just look at the embryo and know what stage it's at. So you can kind of figure it out from the data. Anyway, we did a lot of tricks, and at the end of the day, we got the data. So this is what the data looks like. For each of the 10 species, we have the developmental transcriptome. So in each of these panels, what you see is a bunch of rows where each row corresponds to the gene expression profile of a particular gene. And blue is low and red is high. And you can see many genes are expressed at the beginning, some genes are coming on transiently, then coming down, some genes are coming on later, then staying up. And all species have a dynamic transcriptome. And now you can use this data to study gene expression across the different phyla. So you could take a gene, and say, okay, this is the expression profile, that's nice, but I want more. I can identify the orthologs. You can map the orthologies because we have the sequence, and now you can see, okay, this gene, this is what its profile is like in all these species. And if a particular gene has a particular profile that's really conserved across the 10 species, that's pretty useful too. It's a good resource, but let me show you what we found studying the data. So as an example, let me start with a pair of species. Here's the tardigrade and C. elegans, and we're going to compare the transcriptomes. So what we're going to do is, for each box within this matrix, we're going to compute how correlated is the transcriptome of tardigrade at that particular stage with C. elegans at that particular stage. And this is what we find. So what you see here is that the top left is red. That's because the early C. elegans transcriptome turns out to be very similar to the early tardigrade transcriptome when you compare the orthologs. And also the bottom right is red, which means that the endpoint is very similar. Whatever happens at the end of C. elegans is also getting turned on at the end of tardigrades. But what really struck us is that it seems as though it's organized into two modules. There's the early, and then there's the late. And between them, there's a radical shift, a radical transition, we called it. And it's not something that we just see in this pair of species. We actually see it in all the pairs. So here you see every pair across the 10 phyla. And you can see, okay, if I compare zebrafish with Platinaris. So Platinaris, let's see, zebrafish, and Platinaris, that's this one. You see, it has the early module and then the late module. And I don't know, if you take Drosophila with Nematostella, again, you see this kind of two-module process. Sometimes there's a third thing at the beginning, but that turns out to be that there's a strong maternal deposit. But I think that's really an exception. And that's really, I would say, a maternal phenotype, not an embryonic. So overall, you have something that I think is unexpected. Why should all these species have the same general architecture of building themselves? Like, first you do one thing, then you switch and do something else. To study it further, we asked which genes are these? If there's an early module, which genes are in this early module? And one term that kept on coming up in terms of the gene ontology analysis were cell cycle, chromatin changes, regulation of gene expression. Basically, they're doing mitosis. They're making more cells at the beginning, not surprising. And at the end, if you ask which genes are coming on at the late module, well, these genes tend to be cell type specific, like myosins for muscle, transporters for the nervous system. Again, not surprising that at the end you have things that are more specific, right? Because a tardigrade is going to build a muscle, and C. elegans is going to build a muscle. It's maybe the same muscle. But in the middle, if you ask which genes are coming on at the transition, just as the switch is happening, these are genes involved in signaling pathways and in transcription factors. So when we sent this out, we got back reviews that said, you guys are total idiots because this is really not surprising because everybody knows that in development, first you make cells, then you tell them where to go, and then they differentiate. That was known. And that's true. I would say it's a positive control. But what we really wanted to point out is that now we have something novel, which is we could take these three sets, the two modules and the transition, and map them back. And this is what it looks like when you map back. You see, you can say our worst dream didn't come true. Because what was our worst nightmare? It would be that we had gone to all this effort, and then we have all this data, and we have no idea what to do with it, because how can you compare a sponge developmental transcriptome to a flatworm developmental transcriptome? There's no stages that are comparable. But actually, this gave us a natural way to align them. How? We know there's a switch. They all have a switch. So a switch is like this anchor. It doesn't come at the beginning or the end. It's surprisingly coming at the middle. And now we know where's the early, where's the late. And in 3 of these species, remember I told you that there was a phylotypic stage that was defined at the molecular level? And also when looking at the embryos morphologically. So that's where the switch is happening. In C. elegans, the switch is happening at the ventral enclosure, right where we had mapped it. Also in chordates and in Drosophila, that switch is happening where the phylotypic stage, remember this constriction of variation, that's where it's mapping to. And now in species that no one thought to ask yet where the phylotypic stage is, like in sponge, like in ctenophores, like in Cnidarian, we have a simple way of doing it. We just look at the transcriptome, where's the switch, here's your phylotypic stage. Okay, and then you can say, well, what's exactly happening at this phylotypic stage? And here we found something that really surprised me and that I was trying to deny for the longest time until I realized it's actually interesting. And if you look at which genes are coming on at the transition, in each species, it's a different signature. For example, if you ask which transcription factor pathways are coming on in each species, so what you see is that hedgehog is really strong during the C. elegans mid-developmental transition, and also in echinoderms, and also in the sponge, but not in the other ones. In the other ones, they have a different pattern. So if you look at which pathways and which transcription factors are coming on at the transition, it's different in each species. And this is just signaling pathways and transcription factors. If you ask overall, this is an overall analysis. I won't get into the details exactly how we did it, but what you see here is that the genes expressed in the early module and in the late module are more conserved across species, paradoxically, than expression in the transition. Actually, the mid-developmental transition of each species is more variable than the adjacent modules. So really, this leads us to suggest an inverse hourglass model. When you compare two species across phylum, a summary of the variation looks like this. The early module and the late module is actually similar. They both need to make cells and differentiate. But the middle part, that's different. And how does this gel with what we said before? Because we said before that when you take species, you have an hourglass model. Does this contradict it? I would say no, it does not contradict it, because in the hourglass model, we took a set of species that we proposed to be a part of a phylum. Right? And members of a phylum may have the same body plan. That's what's supposed to be the definition of a phylum. And maybe this is the signature of a phylum. When you now compare across to different species, you see that the variation is in the middle. So what we propose in the paper is that maybe this is the best definition, actually, of what a phylum is. Before, as I showed at the beginning, a phylum is just a branch in the animal tree of life. And you have really very difficult ways of saying, which tree is more of which branch is more of a branchy branch than any other, right? You could say body plan, but how do you exactly define body plan? So here we have a molecular definition that could be objectively tested, that a phylum is a set of species that exhibit this variation when you compare the species within them, yet when you compare it to species not in the group, they will have this concentration of variation in the middle. Okay, so what have I showed you? I showed you that we characterized 10 developmental transcriptomes. I showed you that a universal characteristic of all the species we looked at, and maybe then of all animals in general, is that they have two modules. Their embryogenesis is really two general models. Early phase, a late phase, and a transition that's enriched with transcription factors and signaling pathways, presumably telling where the cells should go. And that we, from this, infer a definition of phyla, and this may allow us to re-examine phyla that were previously defined, but actually there may be sets of phyla. And maybe two phylum that were distinct, that were distinct before, can now be collapsed. This can lead a new path of understanding what a phylum is. And I think in terms of our next directions, we're going to try to work out exactly which networks are operating at the mid-developmental transition. So I want to thank the people who did the work. These are not all the people in the lab now. They're all the people in the lab over the years. In particular, I already highlighted Michal and Leon. And these are all the members of my team over the past 7.5 years at the Technion. Harel is going to give a talk. Where are you, Harel? Here you are. And I want to say that starting next month, I'm actually leaving the Technion and starting a new institute at the New York University School of Medicine. It's going to be called the Institute for Computational Medicine. And one of I think the main kind of goals of the institute is to allow for exploratory science. So it has a very strong computational aspect with big data integration that will, in addition to cool Evo-Devo questions, I think will also address as an institute cancer, genomics, microbiomes, and adaptive immune systems. And I also want to tell you that as I've been trying to transition to NYU, I was on sabbatical, and along with my good friend Martin Lurcher, we wrote a popular science book called The Society of Genes, and I just want to let you know about it. No pressure. But I think it's a nice idea because I got into this field because I really loved the book The Selfish Gene. I was a computer engineer until I read The Selfish Gene, and having studied genomes for a long time, we came to see that a better analogy is, or I would say, of course, they're all selfish genes, but a better analogy for the human genome, because now we're in the age of the genomes. Richard Dawkins didn't know of a single genome in detail when he wrote his book 40 years ago, is that it's actually a society of genes. And these society of genes are competing with each other, quite surprisingly, but they're also cooperating. And that's perhaps the best analogy to understand what the human genome is. All right, so thank you very much.
Thank you very much. That was fantastic. So questions, please.
Yeah.
In your pairwise analysis, did you look at all genes where there were orthologs between those two, or were you only considering genes that had clear orthologs in all 10 species analyzed? Yeah. And the second question would be, do you see at one point in this developmental path a burst of expression of genes that have no orthologs in the other?
Good. Okay, so two related questions on orthologies. Okay, so you might ask in general, why did I present an analysis on pairs? Why not just do a full analysis? So if you ask, I used to work a long time with C. elegans and C. briggsii, and we called them one-to-one orthologs. And then we worked on 5 species, and the one-to-one to one-to-one, it goes down. So it turns out if you want one-to-one to one-to-one to one-to-one to one-to-one, whatever 10 is, you're left with about, it's like 550 orthologs. It really drops down from 20,000. When you do that analysis, you still see the 2 modules. You see them very clear, but when you take pairs at a time, you can analyze 4,000, 5,000 genes, and the signal becomes much stronger. So in the paper, we show it on all, all different kinds of sets, and you see it nonetheless.
This was pure pairwise.
What I showed was pairs, so it's like all 5,000 genes that are together. And then, yeah, so it's kind of odd. I should have pointed out, when you look at the different pairs, each subset is on a different set of orthologs pertaining to just that pair. Okay, and then your question about the orphans. So the truth is, we didn't do it. We didn't study that yet. These data sets are so big, and I don't mean to show off. I'm just really telling the truth. It's a big data set, and you just can't study every single question. So my strategy with these data sets is just to find one cool result and publish that and let everybody else download the data and do their own analyses. So I encourage others to ask, are the non-orthologous genes peaking at different times? And I guess your hypothesis would be that it would be at the same time, though different genes maybe? Or you're going in agnostic. Okay, which is fine. That's fine too.
At the transition.
It's a transition.
Or they might be off at the transition.
Yeah. One reviewer suggested it, and I was like, why don't you do it,
So you've shown, or the data shows a phyletic distinction between phyla, but within phyla we have things we call classes. So inside arthropods, chelicerates, etc. Within nematodes, chromodoria, inoplea. Do you expect the same sort of transitions there? I mean, so within the arthropods, there's enough data now to do that, I would presume, and maybe also within the nematodes, there's enough data to do that. Because one would expect that, so phylesis at the phylum level happened several times. There are also phyla that are more related to each other, like Lophotrochozoa and Ecdysozoa. So evolution is an ongoing process, and these deep splits continue to happen. So it's not clear to me that the deep split in this phyletic transition should be restricted to the origin of animal phyla.
Wow. Okay, that's a very good comment. Let me just summarize it in my own words. Tell me if I've got it right. Really, you're saying evolution doesn't care about, a phyla. There's multiple hierarchies. The hierarchies can be as resolved as we want them to be, because really it's just a branching tree, and it's arbitrary where we cut it. And if we take, let's say we assume I'm right, and the arthropods have this transition. Now you go in and you say there's different classes. Would each class have a different kind of, like a babushka set? You go in and now it's like even more. I don't know. I can tell you that this data is not very resolved. There's a lot more work you could do. You could make it temporally much more resolved. You can go in to look at different cell types, maybe go at every individual cell. Maybe transcription is not the right. So yeah, go ahead.
You could do an Ecdysozoa versus Lophotrochozoa versus Deuterostomia transition on your data.
Right, and I wonder if it's going to be right. If you take Lophotrochozoa versus Ecdysozoa, is it going to be maybe in the early module, because it's kind of like it's not something that's happening in the mid-developmental transition? Maybe it's happening somewhere else, or I think these are all. I think there's so many questions that lead from this that I don't know where to start. Yeah, I mean, let me just say one thing though. I think one kind of resistance that we've gotten from this work is that it may actually help the creationists. Why does it help the creationists? Because creationists love to believe, if I may take it this direction, because the creationists believe that all phyla were spontaneously generated in the Cambrian. Even those that believe in the Cambrian. Okay, so so then, if we're showing here that indeed there's evidence for phyla, because each phyla has a signature, then that's from one kind of view seems anti-evolutionary. I don't see it as anti-evolutionary at all. What I see it is that there could be a process that's really important developmentally, and that process then comes under very strong purifying selection, so strong that no mutation can change it anymore, right? And then if it's so strong, you can have many different kinds of species that are different because of things that are happening before or after, but that particular thing is unchangeable anymore. It's like a frozen accident. I think that would be a definitive aspect of a phylum. I think there's nothing anti-evolutionary about it. Just like, I can give you one example of that, like Hox genes. There's no way you can take out our Hox genes. You just can't do it, right? You can't do it. It's stuck. It's such a crucial part of building us. And is that anti-evolutionary? I don't think so. There could be steps that are frozen and then are not up for gradual change anymore. Yeah. Do you have a question?
Yeah, so, this probably is really stupid, but
I'm sure it's really stupid.
Where does the hourglass...
Just kidding.
Where does the hourglass come from when you look within the phylum if the external hourglass is coming from the genes that are conserved? So is that variation then in genes that are conserved only within the... So is all the hourglass within the phylum coming from genes that are only conserved within the phylum?
Right.
And if you look at the ones that are conserved, do they just flatline through the whole thing and you don't see any difference?
Right. So your question is, how does it make sense if there's very few differences in the hourglass? How do they get magnified when you now compare to different phyla? And I wrestled with this a lot too. And I think that the way to think about it is that even though they're shown right next to each other, there are different scales. So of course, if you compare species that are very similar to each other, there's a lot less differences than if you compare now, Drosophila and C. elegans. So it's a totally different scale. And because it's a totally different scale, the variation is distributed differently. But I admit, it sounds paradoxical, but it's...
You got the data from your earlier study with the
right.
nematodes, so did you try to look at those genes?
Right, so those very same genes that across the 5 species are conserved. If you take those very same genes that are exactly doing the same thing in the mid-developmental transition, you now compare with Drosophila, they're doing something totally different in Drosophila. Patrick.
I just want to follow up on that because I think there's a connection between Mark's question as well. As we get more of this data, you can sort of start thinking about this hierarchical linear model kind of approach where you're really asking what genes are variable within versus between. Are they fixed between?
Yeah.
And then can we, if you put that a phylogeny on top of that, does that create a structured model that then allows you to sort of partition the total variation in these expression patterns sort of at a phylogenetic scale and then at different levels within the phylogeny, and there'll be different genes that emerge that sort of define different transition points and that kind of thing.
Right.
Just a comment, with elegans we have the beautiful RNAi and other lethal phenotypes.
Yeah.
So you can actually ask for each of the genes that are in particular parts of there, how many of those are lethal to C. elegans at that time in its life? And if they're lethal to C. elegans at that time in its life and they're not part of this shared ortholog set, then that's a novelty that elegans has evolved sometime since the Cambrian to stick into whichever part. So if you go to the hourglass part and there's a lethal in there, which is not an ortholog, then that's a novelty that's playing a role in the
I like that. We didn't do that particular analysis. What Mark is suggesting is for every gene, to superimpose upon that whether it's lethal or not. In the 2012 study, we asked for each stage, the genes that are coming on this stage, how lethal are they? And we saw that the ones that are at back then we didn't know about that transition, we'd call it the phylotypic stage, those tend to have a higher fraction of genes that are lethal. But now we could, like you say, fine-tune it into genes that are conserved and not conserved in other phyla.
So we'll have to, one last question, we'll have to move to our next speaker. I'm sorry to interrupt.
Yeah, I'm just wondering, have you paid attention to the maternal to zygotic transition timing, which is coincide with your timing, like your glass bottle or not?
Oh, okay, good. So how does this correlate with the maternal transition? So do you remember I pointed out that the planarians have this like a third module at the beginning there? That's where the maternal to zygotic transition happens very early on. It happens, because that's when the zygote turns on its own, uses its genome for the first time. And what we're talking about here is it's much later in development. So the mid-developmental transition happens much later.
Thank you very much. Sorry.